Good morning. Thank you very much indeed to the organisers for inviting me to this wonderful conference. I'm really enjoying myself. This green oasis is nowhere near as pleasant as the green oasis we're in here, but this is Aston, which is in the centre of Birmingham, and that's where I'm based. So people have already spoken very eloquently and introduced the concept of Alzheimer's disease and the clinical symptoms and how it can be assessed. We're interested in trying to understand the relationship between systemic disease and Alzheimer's. And there's a few comments up here, but perhaps if I can just draw your attention to the questions. And these are some of the real challenges that are out there. We know Alzheimer's disease is the most common form of dementia. We know its incidence is associated with systemic metabolic disease like diabetes and hypercholesterolemia, but not everybody will get the disease. So the big challenges really are who will be more susceptible to the effects of systemic disease, and that's where the biomarkers, I think, start to come in, which we've heard about. And then when, during the pathway to Alzheimer's, is systemic disease important? And again, that relates to when we might be able to intervene in order to delay the onset, perhaps, of dementia. So I guess underpinning all of that is what underlying mechanisms perhaps might mediate the relationship between systemic disease and dementia. So that's kind of illustrated in this cartoon on the side. And actually, inflammation, oxidative stress, and metabolism are all key factors that potentially link the deficits that we see in dementia with some of the systemic diseases. So here we go. If we just look there, we can see that there's been a number of studies which have suggested that actually the decrease in prevalence of Alzheimer's disease is actually associated or can be associated with the use of cholesterol metabolizing inhibitors. So actually, if we use things like statins or anything that will interfere with cholesterol metabolism and reduce cholesterol levels, we can see a decreased prevalence of Alzheimer's disease development later in life. And it was that kind of study that started us moving along this line of looking at systemic metabolism. But let's just take a little bit of a history through some of the micronutrient data that we've also identified. So what you can see here is some of the earlier work we did in collaboration with Christina Polidori, who visited my lab to understand more about oxidants and antioxidants and some of the damaged products that we can find in anybody really, but we were looking in her case at patients who had either Alzheimer's disease or what she termed at the time vascular dementia. And up here we've got a couple of structures. So this is an amino acid tyrosine that's become cross-linked to form dityrosine, and that's an oxidized product, which is very stable. This one here, glutamic semialdehyde, is another modified form of a normal amino acid, but that then has a new carbonyl group on the end of it, just here, and we can measure that carbonyl content. And you can see there wasn't really any mega changes, but there was a tendency or an increase in protein carbonyls in the whole of the plasma or the whole of the serum in patients that had Alzheimer's disease. There was, in fact, a significant increase in the amount of dityrosine in patients that had Alzheimer's disease. And when we looked at this general marker of lipid oxidation called malondialdehyde, in fact, we didn't see anything. If anything, the levels were lower in patients with Alzheimer's disease, which was really intriguing because we didn't expect to see that. So that tells you something about the oxidant story. And this is where we were in 2004. So we think there's some level of oxidation potentially going on in patients who have dementia. In the protein side here, not so much in the lipids. The lipids that was really odd. So if we start then to look at the antioxidant measures that we looked at in these same population, you can see here we have got a number of carotenoids, lutein, zeaxanthine, beta-cryptoxanthine, lycopene, and alpha-carotene and beta-carotene, but all of these ones that are in the orange box here all show to be significantly reduced in patients that have Alzheimer's disease and vascular dementia. So vitamin E is in here, as well as many of these other lipophilic antioxidants. So because the lipophilic antioxidants are clearly going to associate with the lipid components in plasma, like the LDL and the HDL, we thought it was interesting to then follow on and have a little bit more closer look at what was going on with the lipoproteins. So we subsequently isolated LDL from the plasma, and so now we're no longer looking at the total plasma, but we're looking very specifically at the lipoprotein component. This again was pulled out of that data where the epidemiological data suggests that if you reduce cholesterol in midlife, then you potentially reduce the risk of dementia development. When we start to look at the oxidation, again, of the protein, you can see that there's an increase in this level of protein oxidation in the LDL now. So this is very specifically the ApoB. And if we look at then whether that LDL can actually be removed and how effectively it is being removed, we see that it's a really effective scavenger, and it's really very much being taken up by the tissues, and so it's very likely to be creating an oxidative stress in the environment. So we wanted to look at a little bit more at the lipids in these particular LDL. And so what we chose to look at now was isoprostane, which is a more specific and stable measure of what's going on in terms of lipid oxidation. The malondialdehyde data I showed you before is something which has also been suggested to be influenced to an extent, perhaps, by some of the inflammatory prostaglandins. So this one is more specific, and you can see that in the patient group that are now termed AD+, this is a new cohort, and the definition of vascular dementia or AD with vascular symptoms is moving forward a little bit, so it's now termed AD+. So these are AD patients who have symptoms of vascular disease. And you can see here also, if we look at hypercholesterolemic patients who are actually only around the age of 50, mean age is 50 here compared to the mean age here of nearly 80, you can see that there's a very high level, really, of these isoprostanes in people in midlife as well. If we look at how this LDL migrates in a gel, this is just another way of looking at other properties of lipid oxidation, and essentially you can see that patient LDL migrates faster through this gel and further through this gel than the controls. That's true of AD patients, and you can see that's also true of the vascular dementia patients. So we think LDL oxidation is happening in people with hypercholesterolemia, and we can see it's also happening in patients with dementia. It clearly shows that these measures aren't going to be discriminatory at this particular point, but it does suggest there might be something interesting to follow. So our question then is, does LDL oxidation link systemic metabolic disease with dementia? And the way that we've gone about trying to look at that now is a mechanistic study. So I'm going to move away from the clinical data for a little while and just have a look at some of the studies that we've been doing. And the reason that we've started to look at this is to understand why this systemic relationship might exist with diseases in the brain. So if we just think about what's going on with cholesterol, the metabolism is very distinctive. And there is a pool within the brain which is being metabolized independently of the pool which is systemic. And actually there is a potential, though, for some molecules to transport through, either actively or passively, from the blood here into the brain compartment. And there are specific transporters that will take certain molecules through. So, for example, here we've got glucose and amino acids, which will be required by the brain for development. Of course, there are specific transporters for those, but the lipid-like molecules are lipid-soluble and are likely to just shoot through. So all of those are potentially things which mean that it's important to see what's going on peripherally, because it could influence what's going on centrally. So the way that we've established this is to develop a blood-brain barrier in a dish, and then here we're growing the endothelial cells, which are the cells that line the blood vessel. And underneath that, we've got here some astrocytes that we're also growing, so we've differentiated those in culture for many months to introduce this astrocyte-like phenotype, and that forms something like a tight junction. And we can evaluate this tight junction in a series of little tests, which we show here. So these little chopsticks are measuring the electrical current that is allowed to pass between the two chambers, and if the junction here is tight, then when you put an electrical current, there will be resistance to measuring that electrical current in the other field. What we have here also is a way of being able to see how big the pore size is by adding a fluorescently labelled molecule and seeing how much of it moves through. And so if we look at that data just down here, this FITSI refers to the yellow compound here that we're going to look at measuring and see how much comes through, and this transendothelial resistance. tells us something about the difference between the electrical inputs and whether it can actually be measured on the other side. So here you can see that as we culture our cells, when we get to about 14 days, we get a very nice tight barrier. We get increased resistance between the top and the bottom, and we also see very little transfer of this yellow dye through to the bottom. So that's the model that we use. We grow our cells for a couple of weeks in these transwell dishes to get a tight junction. So there we go. If we then start thinking about how might this have any impact and how could we study this in a clinical context, we've done the following. We've taken LDL from a series of subjects, and then we've looked at what happens with that LDL when it's been extracted from the lipids, been extracted from the LDL, and we've analysed those by mass spectrometry, and we've also put those on the barrier to see whether or not they influence that barrier function. Also, we've measured whether or not they influence the oxidative stress inside the cells at the barrier. We've measured whether they trigger inflammatory marker release, and whether that release is going apically here back into the blood, if you like, or basolaterally here down into the brain compartment. So what sort of subjects have we looked at their LDL from? Well, this describes a statin intervention study that we've looked at. So this is essentially saying if we take people who are 50, who are at increased risk potentially of developing dementia because they have high cholesterol in midlife, then what happens if we measure the LDL oxidation, and what does that LDL do to this barrier? So we took these subjects and we had 10 normal cholesterolemic patients, and then we measured the cholesterol, genotype, LDL modification and homocysteine. We then followed them up to 3 months and did similar measurements. This group here received 40 milligrams of simvastatin. They were recruited when their cholesterol levels were more than 6.5 millimolar. It became incredibly difficult to find those because, as somebody was alluding to earlier, intervention is really very rapid, and as soon as cholesterol levels seem to be elevated, statins are often implemented. So we then went to have a look at what the impact of the lipids extracted from these patient LDL would look like. And you can see here that if we look at the untreated samples and then we compare it with healthy controls, there's not much change in barrier permeability, but by the time we get to, and we look at the transendothelial resistance in particular, you can see that the lipids from patients were actually much more likely to cause a loss of that tight barrier, tight junction that we were observing before. Because we think the lipids might be influencing that barrier function because they're causing an oxidative stress inside the endothelial cells, we went on to have a look at what the glutathione concentrations were inside the cells as a measure of oxidative stress. And in our patients, not only was there a significant loss in this barrier, but there was also a significant loss in the glutathione of those cells. We wanted to see what was happening to the barrier, and this is just some fluorescence microscopy that may or may not be clear to you at the back, but essentially there are rings going around the cells here. These are the endothelial cells growing on the wells, and these are tight junction proteins which are formed abundantly around the edge and make sure there's a tight seal. When we add the LDL lipids from healthy people, you can still see these tight barriers are there, but when we start looking at the patient LDL, there's far fewer of these tight barriers. And so that potentially is one of the reasons that we think something could be happening. The blue is the nuclei of these cells, and they still stay pretty tight, so that suggests to us, again, the cells are still viable. They're not dying, but they're just becoming a bit more permeable, a bit more leaky. So we then wanted to see whether or not that influenced the function of those endothelial cells and what would happen in terms of the inflammatory readout. And so we took measurements from the basolateral compartment here and measurements from the apical compartment. And if we look at this inflammatory cytokine, interleukin-6, you can see that both lipids from healthy controls and from patients with high cholesterol led to a significant production of interleukin-6. And this was mainly into the basolateral compartment, which is the brain, if you like. If we looked at TNF-alpha, which is another of the pro-inflammatory cytokines, then you can see again that there is this pattern here of increased secretion into the basolateral compartment, but again, that TNF-alpha is being much more secreted by the lipids that have been extracted from patients who have hypercholesterolemia. We then moved on to see what the effect of the simvastatin treatment would be, and to see whether that would influence the outcome. So again, we're measuring at basolateral, the brain compartment, or the apical, which is the blood compartment. We're measuring interleukin-6 here, and we're measuring TNF-alpha here. And you can see that there is a slightly higher level of production of interleukin-6 in our patients, lipids from the endothelial cells, but there's no difference after the statin intervention in terms of interleukin-6 production. If we looked at TNF-alpha production, again, significantly more TNF being produced by the lipids from the patients pre-statins, but then after statin intervention, that inflammatory effect has been removed. So that suggests to us that the inflammation, there's some kind of inflammation that's associated with LDL in patients who have hypercholesterolemia that may be ameliorated by statin intervention. This here then shows what happens in terms of barrier function, and you can see in our patient group here that, again, this is where there's a significant deficit in the barrier between lipids from control people who haven't got any hypercholesterolemia and lipids from patients with hypercholesterolemia. And after the statin, there's no difference. If we then look at the glutathione, which is our measure of stress inside those endothelial cells, again, you can see this is what I showed you before. There was a significant effect in the patients, that they were significantly lower, but actually when we measured both controls and patients after the intervention, the glutathione levels were unchanged from the healthy controls prior. It's interesting that the data there is much tighter than it was, so we have no explanation for that at the moment. So what's happening to the oxidation of LDL? Well, I showed you before that we had some isoprostanes that were higher in patients. And then if we look at the protein oxidation measurement here, again, you can see it's higher in patients with hypercholesterolemia. After the statins, these levels are pretty much brought back down to normal. So the statins are doing something in midlife that potentially might be useful. But this is all about people who don't have dementia. How does this relate to any of the lipids that are present in Alzheimer's disease LDL? Well, we did a similar experiment where we looked at the effect of lipids on the barrier. Well, first of all, we looked at the isoprostane levels in the Alzheimer's disease patients and the Alzheimer's disease plus with vascular component. And you can see here that the isoprostane level is higher. You can see here, hopefully too, as well, that in Alzheimer's disease and in Alzheimer's disease plus with vascular disease, there is a significant depletion of glutathione in the barrier. So that again suggests oxidative stress from those lipids, more so from those that have a vascular component than from Alzheimer's disease in general. If we then look at what is the effect of these lipids on the function, the barrier function, you can see we're measuring it here again, apically and basolaterally. The basolateral levels of production of this one here, which I think is IL-6, but it's not telling us here that. This is IL-6 production, which is significantly increased in AD and AD plus LDL compared to the control LDL. And then here, this is the permeability. And again, we see some permeability being induced by the healthy control LDL, but actually this is much higher again from the LDL that's come from AD and AD plus patients. So we wanted to try and understand what contributes to elevated levels of oxidized HDL in dementia. Well, this is just a schematic, really, of the complex biology that's going on. It actually highlights some of the genes that were mentioned yesterday in terms of the association with development of dementia and carotenoid deficiencies. And here you can see some of the reverse cholesterol transport molecules that are responsible for taking cholesterol from tissues and removing it out into the liver. And our hypothesis is that we're going to get more of this kind of, more of the LDL if our HDL is less effective at removing cholesterol from tissue, because we presume then the cholesterol is less able to be taken up by cells because they're pretty saturated. And that means the LDL will hang around for longer and have more chance to be oxidized. There's a reason why I've got HDL here, and that will become apparent in the next slide, because when we looked at our dementia patients, we actually saw that their HDL levels were significantly lower. There wasn't a major change in the LDL levels, but the HDL levels were significantly lower in patients with dementia. And we wondered if that might have contributed to that lower malondialdehyde lipid oxidation measure we saw in the very first studies. When we started to look again at the carotenoids, we can see here that we've got zeaxanthin here, which is significantly lower in patients who have Alzheimer's disease with vascular complications. We've also got lutein here being significantly lower, and lycopene also significantly lower. So we suggested that perhaps the removal of lipids by HDL might be diminished in dementia, and we did this reverse cholesterol transport study to see how effective HDL was that was from patients in terms of removing cholesterol. And you can see here again the AD plus patients are much less effective at removing cholesterol. Now I'm just going to go briefly into a supplementation study that refers to tocopherol. Why tocopherol when we're at a carotenoids conference? Well, that's because the data is complete here. What I will then talk to you about after this data has been presented is some preliminary data that we've got from analysis of the cards samples, and that they've been kindly provided by John in collaboration with Howard. So we're very grateful for that collaboration. So when we did a supplementation study with tocopherol, we saw that there was an increase in ApoA1, and that became significant. It was a dose-dependent increase, and it was also time-dependent. It's quite noisy data, but there was a significant increase here within 4 weeks of giving a tocopherol supplementation. And what we're measuring here is ApoA1. This is the molecule that's absolutely key for reverse cholesterol transport. And I'll tell you this now because the tocopherol was, again, another of those molecules, those antioxidant molecules that was at lower levels in our patients with dementia. If we then look at the correlation, we could see that the alpha tocopherol levels relative to cholesterol in our patients, our healthy controls, sorry, again, young people, the ApoA1 levels were directly correlated. So it does suggest that you can influence things in people perhaps when they're younger. And I think that's what our statin study has also shown. In the CARD study, the preliminary analysis we've done is of that isoprostane, which I mentioned before as the stable lipid oxidation product. And what we see, we know isoprostane is inflammatory, we know it can elicit pro-inflammatory cytokines. But the patients, the CARD study then included a placebo randomized controlled trial of Alzheimer's disease patients and controls who received Macular in a 10-10-2 ratio. And whilst the overall antioxidant potential was increased in the overall cohort, we don't know any more than that because we haven't broken the data down yet, the code hasn't been broken, we didn't see any change in the isoprostanes in total. So we haven't been able to influence the overall isoprostane levels at the moment. What we're also looking at in the CARD subjects, and the reason why we haven't been able to break the code yet is because we haven't finished the analysis, is the measurement of oxidized phospholipids. So the Mapstone paper spoke very clearly about there being an association between phospholipids. Our interest is in the oxidized phospholipids because of their potential inflammatory contribution to the development of dementia. At the moment, we're measuring that by mass spectrometry, and we hope to have that data for you all very soon. But our hypothesis behind that is that if you can reduce the levels of oxidation of the phospholipids, we'll potentially reduce the inflammation in the barrier. Remains to be seen whether that's also possible in patients who have overt Alzheimer's disease. So just then to conclude, I think what we've shown is that the LDL lipids from dementia and from hypercholesterolemic patients can cause an oxidative stress in the cell, and we can disrupt the tight junctions as a consequence when we've used zonal occludins 1 as our measurement or as a functional barrier measurement. And we can also see that there's a directional secretion of cytokines which is favouring a pro-inflammatory secretion into a brain compartment. We've also shown that the LDL from the hypercholesterolemic patients is less inflammatory after 3 months of statin treatment, and that is kind of restored then back to the same properties that we see from the LDL in healthy controls of the same age. So cholesterol lowering in midlife has been shown from epidemiological studies to reduce dementia risk. Is there a possible role for micronutrients? Well, we'd like to think there is. We're still looking at that one. And also, could this potentially be attributed to a reduction in circulating oxidized inflammatory lipids? So with that, I'd very much like to thank the various people that have contributed to the hard work in the lab in doing the project, collaborators in Germany, Clinical collaborator is Professor Christina Polidori. We provided our Alzheimer's disease samples, and the Sandwell and West Birmingham hospitals who supported us in the statin study. Thank you very much for your attention.
Okay, questions for Dr. Griffiths?
Very interesting set of data. There's literature indicating that ApoA1 is involved via BCA1 in the uptake of vitamin E. And we recently published that the same mechanism occurs with lutein and zeaxanthin. So that's nice. I think the problem is statin. There's a lot of literature indicating that statin may inhibit ABCA1 and ABCG1 activity. And we also measured that in vivo in animals. So there may be a problem with statins decreasing LDL, but also decreasing antioxidant uptake, and this may pose a problem. What do you think?
I think you're exactly right. And in fact, there is some concern as well about what the long-term risk for dementia might be in patients that take statins. So statins may not be necessarily a good example because they have a whole pleiotropic series of effects that nobody really understands. And I think that is a worry. And I suppose it comes back to the fact that sometimes we find these drugs without really knowing what they're doing. And so a drug perhaps that is good at reducing cholesterol, it's that exercise again. I think that's probably the best drug, isn't it? That would actually then not, in fact, that will raise the HDL, that will increase the bioavailability of micronutrients to cells and potentially reduce the inflammatory properties, actually is perhaps the way forward. Thank you.
So I have a question on the portion of the phospholipid molecule that's being oxidized. Is it the fatty acids? Is it phosphatidylcholine?
Okay, so we're looking at the lipid component that's being oxidized. So it's an lysoxy phosphatidylcholine compound that we're looking at. And it's been shown that those molecules do act via toll-like receptors and induce inflammation in cells. So that's why we were particularly interested in those. We think that the effect of isoprostanes is potentially because they're getting into cells, causing a local inflammatory oxidation and changing redox state and then inducing secretion, whereas the effect of the oxyphospholipids is very much likely to be through toll-like receptors and an active intervention. So.
The question was, is it the oxidation on the fatty acids?
Yes, it is. Yeah. Thank you.
Excuse me. Quick question. Yeah. What form of vitamin E is being given? Is it mixed tocopherols or is it alpha tocopherol?
It was alpha. It was alpha.
Alpha. And then, have you noticed any negative effects of the statins as far as influencing things like CoQ10 levels? And because there's a lot of negative reports that are associated with statin use.
No, you're absolutely right to highlight the negative risks as well associated with statin use. We haven't measured CoQ10. Our outcome measures were very specifically around the lipids, and so we haven't looked at that. But I think that's an interesting point, actually, because that also highlights mitochondrial metabolism. And there's a really interesting growth of study in that area about whether or not some of the shift that we might see, the inflammatory shift we might see, might be in part related to altered metabolism.
How come you use, with the side effects that we know about statins,
why did you use statins as an anti-inflammatory rather than using, say, omega-3s?
We weren't trying to use them as an anti-inflammatory. Our goal was to lower cholesterol. And at the time that we started to do this study, there was less negative press about statins than there is now.
Because there's some indication that actually lower cholesterol in older people actually increases the risk of Alzheimer's. So it's really the inflammation part that may be more important.
Yeah, I mean, I think that's a difficult question, isn't it? Because again, it might be that LDL-HDL balance. And we very clearly showed in that the patients who are presenting with Alzheimer's disease and vascular dementia do have lower HDL. And that's where the deficit seems to lie. So I think that anything that affects HDL, which statins will do, then isn't ideal for older people. Yeah. So it was a model system in younger people that we were looking at. So these are people who had just turned 50, in fact they're between 40 and 64, but been newly diagnosed with hypercholesterolemia, and we wanted to see what the impact was of lowering that cholesterol.
Time for one more question.
Thank you very much for your presentation, and I apologize beforehand if you provided the answer to my question during your presentation. Were the lipoproteins that you used from age-matched patients?
I didn't say that, but they were. So in each of the cohorts that we had age-matched controls. So we were looking at people who were either hypercholesterolemic between 40 and 64 or not hypercholesterolemic, and it was a very simple binary decision-making. And when we were looking at the dementia patients, again, I think actually in the cohort, I don't know if it was significant, but they were perhaps slightly older with dementia, but I don't think it was significant, and the idea was that they should have been age-matched. So, thank you.
Thank you very much.
Okay, thank you.